Today we're going to be talking about cell division. Okay? And cell division can also be referred to as proliferation. And proliferation is usually the term we're going to see in anatomy and physiology. You're going to hear that term often, especially when it comes to tissue growth, tissue development, because as an example, right, so you've got your skin, and let's say you get a cut, right? And so now a bunch of these cells are gone. But you need to replace that, right? Because if you lose your skin, right, you don't want to be bleeding out. And so the cells are going to go through division to fill that space again. I'm oversimplifying. There's a lot that goes on with that process. But essentially, we need our cells to proliferate to be able to grow and to fill the space. So let's just quickly go through the stages of cell division, right? So for most of our cells, right, so the majority of our cells are referred to as somatic, okay? These somatic cells, they are diploid, meaning they have 2 copies of every allele. And so we have of our chromosomes. And so when we divide, we need to do things like double up that amount of chromosomes and then be able to split it so that we have even amounts through. But what we have, right, so I've got my cell, the first phase, we've done all of the things to synthesize our DNA, we've done all of that, and now we're ready to actually go through division. What happens first, right, in our nucleus, okay, is all of that DNA is going to condense, and we can actually see it, which is really cool under the microscope. And when it does that, right, now it's going to be able to be worked with. Before, it was kind of stringy and not condensed. There's not a way for us to easily grab to it. This phase is referred to as prophase. The second phase, okay, is we're going to say this is the whole cell. Now we're going to just go down to that nucleus. Okay, so now we're in just the nucleus. The DNA lines up in the middle of what we call in the middle, and what we call metaphase. So now my DNA is able to be separated. Okay, and the best way I remember the metaphase is M for middle. Okay. And then once the DNA is all lined up, we are able to split them apart. The way we split them apart is by this mitotic spindle that attaches to the chromosomes. Okay, so this is the mitotic spindle. This mitotic spindle has been growing and is now fully established by this stage. Okay. Now we're going to the next phase where we'll be back at the whole cell level, where the 2 parts are being pulled apart. Right? So now we can have 1 copy on each side. We refer to this phase as anaphase. And what's being separated are these sister chromatids. And then finally, once they get all the way to the edge, we're going to start making our new cells. We're going to start reforming our nucleus. Those chromatids can be packaged up again in what we call telophase. I remember telophase because there's 2 cells, and when I'm on the telephone, there are 2 people. This phase, however, is not what separates the 2 cells, it's what's separating the genetic material, which is something that people tend to get confused. This is really just for the genetic material. What separates the 2 cells, right, so right here, You can see this little area, this little pinching area. We refer to this as cytokinesis, which is when the cells are separating. And so those are all the stages of cell division. And when you were going through anatomy, there are going to be times where we're talking about when a cell might get arrested in a different part of the cell cycle, or we'll talk about something that will interrupt a different phase as it moves through.